Hi, it's Ms. Barnhart. Today, let's continue our discussion in genetics by looking at autosomal gene linkage. This is another instance where ratios from crosses do not follow expected ratios. In the first part of this lesson, we looked at genes located on the X chromosome. Now let's look at the patterns of inheritance when two genes or more are located on the same autosomal chromosome. A dihybrid cross between two heterozygotes has a 9:3 to 3:1 ratio. Two different genes that are located close together on the same chromosome definitely do not assort independently because they travel as a pair. We're going to investigate this phenomena in today's lesson. First, let's clarify the difference here in sex linkage and linked genes. Sex linkage refers to a single gene located on the X or the Y chromosome, while linkage, actually autosomal linkage, refers to two or more genes located on the same autosome. Let's revisit the human karyotype. 22 pairs are autosomes, which are homologous pairs. The 23rd pair is different in different genders. Males are XY, while females are XX. Remember, we are going to be investigating how traits are inherited when they are on the same autosome. To see how linkage between genes affects the inheritance of different characters, let's examine another of Morgan's experiments. In this case, the traits are body color and wing size. Wild-type flies have gray bodies and normal-sized wings. Morgan managed to attain through breeding double mutant flies with black bodies and no wings, which we refer to as apterous wings. These mutant alleles are recessive to the wild-type alleles, and neither gene is located on a sex chromosome. The first cross was a P-generation cross to generate F1 dihybrid flies. And the second was a test cross. The resulting flies had a much higher proportion of the combination of traits than in the P-generation flies. We call these the parental phenotypes. We can conclude that these two traits are inherited together because they're so close together on the same chromosome. And we don't see the 1:1 to 1:1 ratio that would be expected on that test cross. However, both of the combinations of traits not seen in the P generation were also produced. We call these recombinant or non-parental phenotypes, which suggested that body color and wing size alleles were not always linked genetically. To understand this, we need to explore genetic recombination, the production of offspring with combinations of traits that are different from those in either P generation parent. Recall that the processes of meiosis and fertilization generate genetic variation among the offspring of sexually reproducing organisms due to crossing over, independent assortment, and random fertilization. In unlinked genes, the recombination is due to independent assortment. You can see the predicted outcome of a test cross between unlinked genes here. Take a look at the cross between a pea plant that is heterozygous for both shape and color, round and yellow, in seeds. When this plant is test crossed with a double recessive, green wrinkled seeds, the following ratio of offspring are produced. Notice that one half of the offspring are expected to have a phenotype matching the parental phenotype, round and yellow or green and wrinkled. We can look back to meiosis to see how this occurs. Consider two traits, A and B, which are located on separate chromosomes. A heterozygous individual would have genotype big A, big B, as shown on these unreplicated chromosomes. Note that the chromosomes are of different sizes, with the contributions of the male parent shown in blue and the female parent shown in pink. The next picture depicts one potential arrangement of how the replicated chromosomes might appear in metaphase 1 of meiosis. When this cell completes meiosis 1, you can see the arrangement is now big A, little b, and little a, big B. This shows recombination can occur because of independent assortment. These two combinations of chromosomes are different here from the parental arrangement. An alternate arrangement is shown here. Can you see why the gametes are predicted to have a 1 to 1 to 1 to 1 ratio? Two of these are in the same arrangement as the parent, big A, big B, and little b, and two are in a different arrangement. They are recombinants, big A, little b, and little a, big B. However, if the genes are located on the same chromosome, then a different pattern emerges. Independent assortment does not occur, and if the genes are close together, then they're inherited as a pair. You would have only two possible gamete possibilities, the parentals. However, there's another factor that causes recombination in linked genes, crossover. Do you remember that crossing over is the exchange of segments of homologous chromosomes during prophase 1 of meiosis? Take a look. Here are the chromosomes prior to replication, just as in the prior example. Now here they are during prophase 1. Can you see how one chromatid is swapping pieces with the homolog? You can see how this allows for a new combination of alleles on one of the arms of the chromosome. Subsequent cell divisions lead to 4 combinations of alleles and gametes. Big A, big B, little a, big B, big A, little b, and little a, little b. These two are the parental combinations of the trait. And these two are the recombinant ones. So let's go back to our flies. If you remember, we crossed a wild-type female with a male with both black body and apterous wings. All of the F1 offspring were wild. This led us to conclude that both black body and apterous wings were recessive. These flies are heterozygous. We then completed a test cross with the F1 hybrid and a homozygous recessive male fly. That is, one with apterous wings and a black body. Using G's and N's to refer to the alleles, you can see that this cross should result in a 1 to 1 to 1 to 1 ratio of offspring if the genes are not linked. If they are linked, then we would expect to see more of the parental genotypes. As you can see, the data do not show a 1 to 1 to 1 to 1 ratio, so the traits appear to be linked. The genes appear to be on the same chromosome. There are many more of the parental genotypes that would be expected if they were unlinked and located on separate chromosomes. So, how does the amount of crossover relate to the distance between the genes? The further apart genes are on a chromosome, the more frequently they cross over. We will indicate that each 1% of crossover is equal to 1 map unit, or centimorgan, of distance between the genes. Using our experimental data, you can calculate the estimated difference between the genes. We calculate the percent crossover as the number of recombinants divided by the total number of individuals, and then we multiply by 100. In this case, this means 37 plus 35, divided by the total number of offspring, which in this case is 1,187. Doing this math gives a 6.1% crossover. Repeated crosses give data that can be used by scientists to generate a gene map of the locations of genes on the 4 fruit fly chromosomes. This gene map is shown here. Here's the location of the apterous wing and the black body mutation. If you subtract the two locations, you can find the percentage of crossover we should expect. In this case, it's 6.7%. Chi-square analysis could be used to determine the validity of this prediction. Now let's take a look at a sample problem from a prior AP exam. Be sure to pause the video as needed to collect and record the data. A new species of fly was discovered on an island in the South Pacific. Several different crosses were performed. each using 100 females and 100 males. The phenotypes of the parents and the resulting offspring were recorded. In the first cross, true breeding bronze-eyed males were crossed with true breeding red-eyed females. All of the F1 offspring had bronze eyes. F1 flies were crossed, and the data for the resulting F2 flies are given in the table below. Cross 2. True breeding normal winged males were crossed. True breeding normal wing males were crossed with true breeding stunted wing females. All of the F1 offspring had stunted wings. F1 flies were crossed, and the data for the resulting F2 flies are given in the table below. Cross 3. True breeding bronze-eyed stunted wing males were crossed with true breeding red-eyed normal-winged females. All of the F1 offspring had bronze eyes and stunted wings. The F1 flies were crossed with true breeding red-eyed normal-winged flies, and the results are shown in the table below. Part A. What conclusions can be drawn from cross 1 and cross 2? Explain how the data support your conclusions for each cross. Part B. What conclusions can be drawn from the data from cross 3? Explain how the data support your conclusions. Be sure to pause the video to work on your predictions. Now let's check and see what you did. You should have identified that bronze is dominant over red and that this is an autosomal trait. Possible explanation is that all F1 or heterozygotes express the dominant trait, which is bronze, and the F2 show a 3-to-1 ratio. We can tell they're not sex-linked because we see an equal distribution of the F2 phenotypes for both genders. A similar conclusion can be made for cross 2. The stunted wing is dominant and the normal is recessive. This is also an autosomal non-sex-linked trait. Explanations are the F1 heterozygotes express the dominant trait, which is stunted. The F2 shows a 3-to-1 ratio. And there is an equal distribution of the F2 phenotypes for both genders. For cross 3, you should have noted that these genes appear to be linked, that crossing over caused the recombinants, with the genes 10 map units apart. The explanation here is that I do not see a 1-to-1 to 1-to-1 ratio, as predicted by independent assortment. The frequency of recombinant phenotypes is 10%. The parental phenotypes, the bronze stunted and the red normal, are represented in 90% of the offspring. I hope that was helpful.